I'm David Castillo, CSO of BatchX. In this tutorial I'm going to show you how to run a structural alteration pipeline using RNA Seq data. So let's go into the Tools and Pipeline section and search for RNA Seq. This will display all the tools and pipelines under this category, and for this tutorial we are going to focus on structural variants. So let's click on that. And now we are seeing the documentation from the pipeline, all the tools that the pipeline orchestrates, as well as a diagram showing how tools interact and the data flows. The documentation also includes inputs and outputs, as well as some examples of how the inputs should look like. The pipeline also includes a usage section where we can see examples and run them without actually having to upload any data to BatchX. There are several examples, and for this case we're going to run an analysis that only detects gene fusions. Even though the pipeline has been implemented to identify other types of variants like alternative splicing, measure splicing efficiency, as well as perform differential exon usage. For this tutorial we're going to keep things simple and run this example. So let's click on Run. This takes us to this other screen where we can configure the job. In this case, everything the platform has already configured, all the inputs, but in the case that you wanted to use this as a template, you basically will have to only replace the FASTQ files. So let's click on Submit and run this job. This other screen shows us the inputs that we have provided to the pipeline, and given that I have run this analysis in the past, the platform is going to identify this and will return the same results we got in the last execution. If we refresh this, okay, no need for that. We're already seeing all the outputs that the pipeline has generated, and for this tutorial we're only going to focus on the report, which contains all the information that the pipeline has generated. So I have already downloaded this file and unzip it so we can save some time. So let's just click on this tab and inspect its content. This section that we're seeing is the quality controls, where we can see the reads and the number of reads that were left after each one of the filtering steps, as well as the distribution of reads depending on the genomic origin. What you would like to see is that most of the reads fall into the exon, right? But it's also common to see some of the reads falling in the intron areas. The rest of the plots shows if there are any bias in the coverage of the transcripts, as well as the distribution of reads along the genes. Right? What you would like to see is that there's an even distribution from beginning to end of the genes, but if we happen to see a dip on the coverage, that might indicate that RNA could be degraded. The same information that we're seeing in the plots, it's also available in the form of tables that you can download. And let's move to the second section with the ideogram. Here at a glance we can see how the reads are distributed under two categories, one being the exons and the rest of the regions shown in yellow. Right? We can see that most of the reads fall into the exon areas, but we are losing some of those on specific chromosomes. Right? We can inspect each one of them chromosome by chromosome and see where we might be losing some of the reads. In this analysis, given that we run gene fusions, we have used two of the algorithms that we have in the catalog, Arriba and Star Fusion. Each one of them has slightly different approaches, but in both cases we are going to see a table with the variants that were identified, as well as links that show us a very nice representation of the variant that was detected. Right? We can here see the exons of each one of the transcripts, we can see how the breakpoint occurred and the resulting gene fusion, as well as the retained protein domain. Going back to the report, in case we have run the analysis including other types of algorithms like the ones I mentioned before, we will be seeing additional sections here. Okay, so that concludes our RNA Seq structural alteration tutorial. Hope you enjoy it, and looking forward to see you in BatchX. Bye bye.